The title of our class session today is Worst Pickup Line Ever. You're brainy and have a great nervous system. So we will be exploring biological psychology, also known as biopsych, with a focus on the brain and the nervous system. And we are going to start with a test your learning on the structure of the neuron first thing. You have 2 minutes for this challenge. A couple of new ones are added in here, and that is beyond your book as a learning tool. So I want to see if you can guess them. Ready? And 2 minutes. Go. Okay, time is up. So let's review these parts of the neuron. Number 1 is the myelin sheath. Number 2 are the terminal branches. Number 3 is the axon. Number 4 is the synapse, sometimes called the synaptic gap. Number 5 is the nucleus. Number 6, terminal buttons. Number 7, dendrites. And number 8 is the soma, or the body of the neuron. This figure was also posted for you in your timeline this last week. We recommend that you download 2 copies of it. This one and another where you have blacked out the answers. Then staple them back to back. And voila, you have a quick, easy way to actively test yourself with immediate feedback. Remember, this is the most effective way of learning, testing with instant feedback. The time that you put in to preparing your figures this way, you more than save when learning them. Neurons are nerve cells in your brain responsible for your thinking. When you are thinking about something, the pattern of neurons firing across the brain is different than when you think about something else. So it's like a dot matrix where the dots are activated and cross the entire screen, but which ones that are activated determine the image. Now, the difference between neurons and nerve cells in other parts of the body is that neurons have these communication parts for spreading thoughts, for communicating, right? The communication parts are the dendrites and see these coming off the body or soma there. The axon, this long tube here, and the terminal branches at the end of the axon. So these parts are unique to neurons. Other cells in your body simply resemble the soma or body of neurons. There are about 100 billion neurons in the brain and about the same amount of glia cells. Glia means glue, by the way, so glia cells are literally the gooey stuff between neurons. An easy way to tell the difference between dendrites and terminal branches is that dendrites come from the body, soma, of the neuron, while the terminal branches are at the very end of the axon, right? But also, see the shape on the ends. If you look at these dendrites coming off the body or soma, you see that they very much resemble antlers, and if you look at the very ends of each one, you'll see that they are somewhat pointed. This is very different when you come over to these terminal branches. I think you can see it the best in these two that I'm pointing my cursor at right now. The end shape resembles a hoof. And these are called terminal buttons or terminal boutons, oftentimes the French pronunciation. Okay, let me point out something important about this figure, which I guarantee will be on your first test. This figure will. So note that the line representing the axon is in the middle of the myelin sheath, right through the center of that. There is a vertical line, right in the center. That is representing the axon. And notice that there are two items right here that could appear on your test that are pointing to different parts. This one is stopping on the outside myelin sheath, so the answer is myelin sheath. But this line goes right to that center line, so that is the axon. Remember that, very important to look at that closely if that comes up on your test. So how do your thoughts spread? The resting potential of an inactive neuron, which is the voltage it carries when it's not actively firing, is a negative 70 millivolts. A negative 70 millivolts, in other words. It's doing nothing. When a neighboring neuron is firing, it can affect the charge of its nearby neurons and can change their charge. If their charge becomes above a negative 70 millivolts, they become excitatory, meaning more likely to fire. But if it changes to below a negative 70 millivolts, it becomes inhibitory or less likely to fire. So when a thought is spreading in your mind, the neurons involved are excitatory, right? But if you start thinking about something else and that fades, the charge in neurons involving the first thought become more negative and inhibitory. So here is a quick quiz question. What if these molecules in these receptor sites change the voltage of the next neuron to a negative 50 millivolts? Would it be more or less likely to fire? Think about it. It's more likely to fire, right? Because a negative 50 is more positive than a negative 70. Now, how about if it changed it to a negative 90 millivolts? It would be less likely to fire, right? That would be inhibitory. Okay, so here is a close-up of a terminal button. We're really zooming in here because remember that terminal buttons or terminal boutons are at the very end of terminal branches. When the charge goes through a neuron, remember that long axon? It comes through as an electrical charge, right? But something kind of cool happens at the terminal buttons. It turns into a chemical charge. And let's see how that happens. So first, notice these chemical sacs in the terminal button here. And notice that that electrical charge is causing them to release into the synaptic gap. Over here, we've got receptor sites that represent the membrane of the next neuron. Okay? So if enough of these chemical molecules come out here and actually fit into these receptors, that is what can change the charge of that next neuron and cause it to fire.